let's talk about the polymerase chain reaction, or PCR for short. Now, this is a very powerful technique, so powerful that a Nobel Prize was awarded for its invention. Now, Nobel Prizes are generally given for a lifetime of a contribution to the sciences or other endeavors, and rarely to a single seminal invention, but PCR is just that, a single seminal invention that had profound consequences for our ability to manipulate genes, to do studies of gene regulation, and to simply produce lots of sequences so that we could study them. So it's a very powerful technique, and the inventor really deserved the Nobel Prize for it. So let's see what this is all about. Let me remind you that, like most of the techniques, if not all of the techniques in gene manipulation, we rely on the tools that we develop by understanding replication. So let's take a look at DNA synthesis in a test tube as we might actually do it to get a sense of how PCR would work. So first, we start with double-stranded DNA, which is going to serve as a template for the production of some new DNA that we would like to get. So we identify a region of interest, a gene, for example, somewhere in the middle of a chromosome that we would like to make lots of. So the first thing we might do if we wanted to make some copies is simply heat the DNA to denature it, to separate it into two strands. At this point, we would add a primer, because if we're going to use DNA polymerase, as you'll see we will in just a moment, DNA polymerase is going to make a new strand for us against one of these blue template strands, but it's going to do that by adding to a primer. So the first thing we have to do is actually know a little bit about the gene we're interested in, and you already saw in one example that we can get such sequence information even if we only know the sequence of the polypeptide encoded by this gene. Nowadays, of course, we get the sequence from many sources. It could be from the polypeptide, or it could even be from a similar gene from some other source where we know that there's a sequence that we should expect to find here. So we make a synthetic oligonucleotide primer shown here and cool the DNA down. We've denatured it by heating it, so if we cool it down, the primer will be able to H-bond to its complement somewhere in the gene and form stable H-bonds. So we have to cool it down to do that. At that point, we will add some additional components that we know are going to be necessary to do replication or DNA synthesis. We will add the 4-deoxynucleotide triphosphates, DATP, DCTP, DGTP, and DTTP. And of course, we're going to add DNA polymerase, shown on the right. Well, when all of that stuff is added and the temperature is just right around 37 degrees, the polymerase is going to find the end of the primer and do what you just saw happen. It's going to extend the strand. I didn't show the polymerase moving, but as you can imagine, it would move along the strand as it was synthesizing it. And so we've made a strand. Now let's say we want to make lots of copies of this new strand. Well, we would have to, after making the first copy, we would have to denature that DNA, that double strand, and we would have to add fresh DNA polymerase simply because when we heat to denature, the heat would permanently inactivate the DNA polymerase. If you heat the DNA polymerase, it does not fold back into a conformation that is active as an enzyme. This is one of the enzymes, in other words, that pasteurization would knock out pretty quickly. So we'd have to add new DNA polymerase. We actually wouldn't have to add more primer or more of the 4-deoxynucleotide triphosphates because they're pretty stable even when they're heated up to pretty high temperature. But we would have to add new DNA polymerase. And if we did that, we could make, if we repeated it 20, 30, 40 times each time, we would make one more copy. And so if we did it, say, 50 times, we would make 50 copies of the new strand of DNA. And as it turns out, that's not enough to do anything with anyhow. So the Nobel Prize that was awarded for PCR was awarded for the discovery of a method to do the following. So the polymerase chain reaction, first of all, is a real time saver. It's an end run around cloning very often. In fact, cloning, while it is still done, cloning of the sort that I showed you, cDNA libraries and genomic libraries, it's really not the method of choice these days to look for and get the gene that you're interested in, the needle in the haystack. In fact, PCR is very much more often the way to do it. So what do you need to do PCR? Well, like every other technique I've shown you, you do need some knowledge of the DNA sequence that you're after. Enough knowledge in this case to make primers. Now in PCR, you're going to make primers for both strands of DNA in the region of interest. I should put region of interest in quotes. That means the gene you want to have a lot of. or the sequence of DNA that you wish to make a lot of. So you're going to need primers for both strands. We call them opposing primers. You'll see that in a moment. And they will be positioned in the DNA when they form the H bonds with their complements at some distance apart. And you'll see what that means in a second as well. And another clever part of PCR was the use of thermophilic DNA polymerases that are not inactivated by heat. The result, and I'll explain that in a moment. The result was that DNA could be geometrically amplified, meaning essentially the amount of DNA that you start with can be doubled every time you do one of these reactions. And you can figure out after 25 or 35 cycles of doing this, we call them PCR cycles, you're going to have very large amounts of DNA. But let's take a look at how this actually works, what we have to do to get PCR going. Okay, so the first thing we have to do is make the synthetic oligonucleotide primers. Let's take as an example something that might really have been done at one time. So here's the sequence of both strands of a known gene in Drosophila. It doesn't matter what this gene might actually code for, but it's a region that has been sequenced. And the end game, the goal, is to see if there's a homologous gene in other organisms, let's say in humans. So here's that sequence. Don't look at it too carefully. The immediate goal is to make these synthetic primers. So what we need to do is identify two sequences that will be opposing primers that a thermophilic DNA polymerase can use to synthesize that part of the gene we'd like to get at. We're going to amplify this new DNA by PCR. So let's make the primers. Let's see what I mean by identifying the primers. I have highlighted in black, in boldface, two sequences. I don't remember how long they are, probably 17 or 18 bases long, each of which might be a good sequence to synthesize to make a synthetic primer. When we make the primer, we're going to synthesize them in an appropriate direction. So the top one, just shown now in red with the arrow pointing to the right, would read GT, GG, AT, etc., ending in TGA at the 3' end. The opposing primer would be read as GC, TAA, TC, etc., ending with CC at the 3' end. So if you think about the geometry of all this, each of these two sequences, if we actually synthesize them, and we do that routinely now, could serve as primers, and they would form H bonds with the opposite strand, the strand just above them, above the one that begins GCTAA, reading from right to left, or just below it, reading GTGGA from the left, going to the right. You'll see what that geometry looks like with a more cartoonish drawing. So before I go any further, the thermophilic polymerase I'm referring to is one that was isolated from a thermophilic bacterium, Thermus aquaticus, called TAC, T-A-Q for short. And TAC DNA polymerase is the most commonly used. And again, the beauty of TAC polymerase, of a thermophilic polymerase, is that it is not destroyed when you heat it up to temperatures as high as 100 degrees. And you can heat DNA in the presence of this enzyme to denature it. And when it cools down so that the primers can bind to the DNA, the TAC DNA polymerase is still alive and well and can catalyze polymerization. So then you only have to add the 4 deoxynucleotide triphosphates to actually get DNA synthesis. Okay, so hopefully that's not too confusing. Let's take a look at actual PCR, the amplification of DNA. We're going to look at the first several rounds of replication, and then you should get a sense of how quickly you can produce large amounts of DNA using PCR. Again, this technique I'm about to show you was really remarkable and prize-winning. So here we have the 2 strands of our template DNA. We're actually going to add the opposing primers we just produced, and TAC polymerase, and the 4 deoxynucleotide triphosphates all at once in a single tube containing the template DNA. We'll heat the DNA to denature it at 100 degrees Celsius, and then we're going to cool it to approximately 45 degrees to allow the oligonucleotide primers, these opposing primers, to form H bonds with their complements on either strand. And here they are, annealed or H-bonded to their complementary 17 or 18 bases or 20 bases, however many that was, in the complementary strands in the region of interest. And remember, these primers were created by knowing some sequence of the region of the DNA that we wish to get a lot of. All right, so the next step is to reheat to, let's say, 70 degrees or so, and that turns out to be the optimum temperature for the activity of Taq DNA polymerase. Now, remember, the deoxynucleotide triphosphates are already there, and the two primers have annealed on opposite strands, and the polymerase is there and it's active. So we're going to get DNA synthesis, and that's shown in the lavender strands that we've just extended in either direction off of the two templates. That's the end of the first PCR cycle. We're going to repeat the cycle of temperature changes. So what that does is it denatures the DNA at 100 degrees. At 45 degrees, the primers can now form H-bonds with their complements, and that's shown here, but now it's happening not on 2 strands, but on 4 strands of DNA, right? And when you bring the temperature back to about 70 degrees, the polymerase, being still active, can extend the fragments. Now, if you look closely, you will see that we've made 2 new fragments in green that are actually quite different than the original lavender fragments, or in fact, than any of the lavender fragments that we made. So we make 2 more lavender fragments and 2 green fragments, both different lengths. The lavender fragments could be quite long, but the green fragments only go out as far as the end of the strand that they're being made against. Now that marks the end of the second PCR cycle. I've not drawn the next or third cycle, but if you imagine denaturing these 4 double strands, in the next round, you are going to make another pair of long DNA, lavender DNA strands, but you're now also going to make the complement of each of the green strands, and they will only be as long as what you see here. Okay, so the asterisked strands, the short ones, are now going to double in the third cycle and each subsequent cycle. What you're really after are the asterisk strands. One more cycle, the third cycle, you'll have 1 double strand about as long as what you see here of the asterisked DNA. In the fourth cycle, you will have two double-stranded pairs, and in the next cycle, 8 double-stranded pairs, and so on. What happens to the long lavender fragments is that you get 1 more every cycle, and they don't contribute very much, but you can imagine if you keep doubling the short double strands now every cycle, you're going to have quite a bit. So the number of target DNA sequences doubles each round of replication after the third cycle. So starting after the third cycle, you get the target that you're really after beginning to accumulate in large numbers. So you can think or have fun doing the calculations to figure out how many copies of a gene sequence could you really get after, say, 30 rounds of PCR, which means realistically you start counting after round 3, right? So round 4 through round 30, 26 cycles. I don't have the answer for you. You can figure it out. But I can tell you the number is, in fact, quite huge. Even though the number of copies is going to be somewhat lower than the theoretical limit you could calculate, it'll still be quite large. You can start with the DNA from a single cell or a single fragment that might be found, for example, at a crime scene, and produce microgram, or if you really want to, close to milligram quantities of that DNA. Now, microgram quantities, that's a millionth of a gram, and it might sound small, but it's actually the amounts of DNA that we typically work with, and if you want more, you can get more. You can get this microgram quantity of DNA in a matter of, say, 4 to 5 hours using a thermocycling machine that programs all the temperature changes in that tube automatically, so you don't even have to babysit the tube. So PCR can get you these large quantities of some single gene in literally no time at all. So, power of PCR. Start with 1 or a few related clone sequences and use these to design primers which will PCR amplify homologous sequences, for example, from the same organism or different organisms. So, for example, why the same organism? Well, you actually have multiple genes for globin, and they are related to one another, and it is possible to PCR amplify each of the different genes using sequences for primers that are common to each of those different genes. Likewise, if you want to isolate kangaroo globin and you only have human globin sequences to start with, you might be able to figure out a set of primers that could amplify kangaroo globin genes, carp globin genes. You name the possibilities, and they are there. You can then, of course, sequence the PCR-amplified fragments that you get to study the evolution of genes and species. Obviously, a fish globin gene isn't going to be identical to a human or a cow or a frog gene, for globin. So you can study the differences between the genes of different species and use that information to make statements or conclusions about species evolution and gene evolution as well. Of course, you can also study the role and regulation of related genes in different species as well, or in the same species. So you can use PCR amplifying genes as probes to study the regulation of actual genes in cells as they are turned on or turned off. We used to do this, by the way, with genes that we had cloned by cDNA cloning or by genomic cloning the hard way, just to give you a sense of the difference. If you know some sequence information, you can do cDNA cloning. When we did it many years ago, even today I suspect, you would be able to do cDNA cloning and get a clone that you want and sequence it perhaps within 3 to 6 months, depending on what you were looking at. Compare that to getting the same quantities, if not even more, of the gene of interest in a day. So that, I hope, gives you an idea of why PCR is so widely used. It's obviously used in molecular biology laboratories for many of the reasons I showed you, and of course it's also used in forensic laboratories. Much of what you see on the CSI television programs often overstate how quickly things can be done. But one thing that can certainly be done quickly is PCR, dealing with the starting bits of hair or bits of skin or whatever that they get from a crime scene. These crime scene investigators can do PCR and then do analyses on that DNA. Those analyses usually take longer than what they tell you, but just getting the DNA is very quick. And that brings us to the end of this presentation.